In 2008, a research group published a study repositioning a drug target for a rare metabolic disease. 3 independent labs tried to validate it. None could. The sequence they deposited in GenBank had a 2-base substitution right in the open reading frame. It wasn't a PCR error. It wasn't contamination. It was a sequencing artifact. The kind that only appears when someone reads a chromatogram like a photograph, instead of understanding what it actually is.
Was the error at the primer binding site?
No. Base 51, right at the edge of the reliable read window. Their quality filter wasn't tight enough. They called a peak that was noise.
It is mildly ironic that the technique designed to read genetic information was the one that was misread.
How much drug development capital was downstream of that sequence?
Enough that 3 labs wasted the better part of 2 years before anyone reran the original sample. The correction ran as a single paragraph in a journal most people didn't read. Here is what happened. The first 20 to 50 bases after any primer are a dead zone, unresolved signal overlapping noise, nothing you can trust. Base 51 is right where the read starts to become reliable. This group's target sequence sat at that boundary, and they didn't check the quality scores. Once you understand how Sanger generates a read, the chemistry, the physics of capillary separation, what a peak actually represents, you will never submit an unchecked chromatogram again. That is what we are building today.
Sanger sequencing, developed by Frederick Sanger in 1977, is built on a single chemical principle. Normal deoxyribonucleotides have a 3' hydroxyl group on their deoxyribose ring. That 3-OH is what the polymerase uses to form each successive phosphodiester bond. Dideoxynucleotides, DDNTPs, are missing that hydroxyl. When a DDNTP is incorporated, extension stops permanently at that position. No 3-OH, no bond, no further chain. In a modern sequencing reaction, all 4 DDNTPs are present simultaneously, each labeled with a spectrally distinct fluorophore. Polymerase extends your template from the primer, and at every position, there is a competition. Incorporate a regular DNTP and continue, or incorporate a DDNTP and terminate. The result is billions of fragments covering every possible length, each terminated at a known nucleotide, each fluorescently tagged to identify it. Capillary electrophoresis separates these fragments by size with single nucleotide resolution. A laser reads each fragment's color as it passes the detector. Time equals size equals position in the sequence. That is your chromatogram.
If this episode has taught you something in the last 3 minutes, subscribe before you forget. Every Monday, one technique, explained by people who have actually broken it. Subscribe. We will be here next week.
A Sanger sequencing run at a core facility costs roughly €3 per sample. That is the cheapest genetic data you will ever buy. The sequencer generating it costs $200,000. The economics of centralized core access are doing a remarkable amount of quiet work.
Right. What you need. Template DNA, and it must be quantified. Not estimated, not guessed. Quantified. For PCR products, 5 to 20 nanograms per microliter in the final reaction. For plasmid DNA, 50 to 100 nanograms per microliter. Sanger runs on linear amplification. There is no exponential recovery from weak starting material. If your template is thin, your read is thin.
I didn't quantify mine.
What did you put in?
An amount that seemed reasonable.
It is mildly clarifying that we now know volume was involved.
We'll find out at the data step. Sequencing primer at 3.2 micromolar. Single primer only. Sanger is not PCR. You read in one direction from one primer. You do not need a reverse primer for the extension reaction. Big dye terminator V3.1 ready reaction. Mix. This is your commercial master mix. It contains modified polymerase, unlabeled DNTPs, and all 4 fluorescent DDNTPs. Store at -20. Keep it away from light during handling. It degrades in ambient room light faster than the product sheet states.
The original Sanger protocol used 4 separate reactions, each with a single unlabeled DDNTP and radioactive labeling for detection. The modern single-tube format was made possible by advances in fluorescent dye chemistry that allow 4 emission spectra to be separated simultaneously by the detector. 4 reactions became 1. Radioactive labeling became optical. 40 years of chemistry, 1 tube.
It is mildly convenient that 40 years of incremental chemistry also eliminated the radioisotope handling license requirement.
Cleanup reagent, exosapity, or ethanol ether precipitation. I will come back to this step and explain exactly why it is mandatory. And access to a capillary sequencer, in-house or through a core facility. You submit a plate, you receive a file, protocol, step 1, reaction setup. Mix 5 microliters, big dye 1.6 microliters primer at 3.2 micromolar, 1 to 3 microliters template and nucleus-free water to 10. Keep everything on ice. Keep it away from light. Nucleus-free water specifically. Not the water bottle on the bench. Not the water from the sink.
I used the water from the sink.
The tap water.
The nuclease-free bottle was empty. The water looked the same.
The water is not the same.
The big dye reaction buffer contains a precisely defined magnesium concentration calibrated for the polymerase's activity. Municipal tap water contains divalent cations, magnesium, calcium, at variable and uncontrolled concentrations. Adding these to the reaction shifts polymerase kinetics unpredictably. You can get truncated products, reduced terminator incorporation, or no product at all. Tap water also carries RNases and DNases at concentrations sufficient to begin degrading your template within minutes at room temperature. Nuclease-free water costs €4 for 500 milliliters. This is not a premium product. It is a controlled reagent.
Step 2. Cycle sequencing. 96 for 1 minute initial denaturation, then 25 to 30 cycles of 96 for 10 seconds, annealing at 50 for 5 seconds, extension at 60 for 4 minutes. The extension is at 60, not 72. Big dye polymerase is not tack. Running it hotter does not make it faster, it makes it wrong.
Mine is set to 72. I used the PCR preset on the thermocycler.
You ran a Sanger reaction on a PCR program.
It's the same cycling. I was saving setup time.
It is not the same cycling. At 72 degrees, the modified polymerase loses processivity on longer fragments. Your read will look clean for the first 300 bases and then fall apart completely. You'll submit it thinking you sequenced 800 bases. You sequenced 300 bases and noise.
The mechanism is the enzyme's thermal stability profile. The modified polymerase in big dye achieves its optimal processivity and accurate DDNTP terminator incorporation ratio at 60 degrees. At 72 degrees C, the enzyme remains active but begins to dissociate from longer templates before completing extension. Fragments above approximately 300 to 400 bases are systematically underrepresented. This failure is not catastrophic. The read does not disappear. It degrades gradually past a threshold, mimicking low template signal rather than a temperature error. You will not immediately recognize it as a thermocycler setting problem.
Step 3. Clean up. This is where most bad runs start. After cycle sequencing, your reaction contains unincorporated dye terminators, free fluorescent DDNTPs that were never incorporated into any fragment. If you inject this reaction directly into the capillary, those free dye molecules migrate faster than any of your labeled fragments, arrive at the detector simultaneously, and saturate the signal. You get a dye blob, a fluorescence burst that overwhelms the detector for the first 30 to 50 bases of the run. Anything under it is gone. 2 cleanup options: exosapity. Add 1 microliter, 30 for 15 minutes, 80 for 50 minutes to inactivate. Or ethanol ether precipitation. Add ether and ethanol. Spin 30 minutes at 3000 gram. Wash with 70% ethanol, dry completely. Resuspend in hydroformamide. Either method. Neither is optional.
I thought cleanup was an optional washing step for very sensitive applications.
It is not optional for any application.
Unincorporated dye terminators are small molecules. They migrate through the capillary polymer faster than any labeled fragment and arrive at the laser detection window before the sequence read begins. Because all 4 fluorescent species arrive simultaneously, The detector sees a multicolor saturation burst, the dye blob. This signal overwrites any real data in that window. Cleanup removes these molecules either by enzymatic digestion, as ExoSAP IT does, or by size-based precipitation. Without it, you lose the beginning of every read, every time, structurally, not by chance.
It is mildly reassuring that the step most commonly described as optional is the one that destroys the most data when skipped.
ExoSAP costs approximately 0.15 euros per reaction. One failed core facility run costs 3 euros. Skipping cleanup to save 0.15 euros is a 4 euro decision made on principle.
Step 4. Capillary electrophoresis. Your core facility handles injection. But understand the physics. Your cleaned, resuspended fragments are denatured to single-stranded form in formamide and injected into a polymer-filled glass capillary under high voltage. Shorter fragments migrate faster through the polymer and reach the laser detector first. Longer fragments arrive later. The laser excites each fragment's fluorophore as it passes. The detector records emission wavelength, which base terminated that fragment, and time, which corresponds to fragment size, which corresponds to sequence position. That stream of color over time is your chromatogram.
The polymer in a capillary sequencer is a linear polyacrylamide-based matrix injected fresh before each run. Unlike the slab polyacrylamide gels used in original Sanger protocols, the capillary format regenerates the separation matrix between every injection. This eliminates the between-run gel variability that limited throughput and reproducibility in early sequencing. Consistent polymer, consistent resolution, consistent read length. That consistency is a large part of why capillary Sanger was the format that completed the Human Genome Project.
Wait, if you have fragments of every possible length all in the same tube, how does the detector know what order the bases go in?
The order comes directly from electrophoresis. Each fragment is a different length, and length determines migration speed. The shortest fragment arrives at the detector first, the next shortest arrives next, continuing in order through to the longest. So the detector reads them sequentially. Fragment of length 1, then length 2, then length 3, all the way to the end. Since fragment length corresponds to position in the sequence, you are reading the sequence one base at a time, in order as the fragments stream through the detection window. The chromatogram's time axis is the sequence.
Good data. Clean, symmetric, well-separated peaks with consistent height. Each peak should have a single apex and a narrow base. Background signal between peaks should be low. Reliable read length of 700 to 800 bases. Fred scores above 20 across the red, above 40 in the mid-red region.
Fred scores encode the probability of a base call error. Fred 20 is 1 error per 100 bases. Fred 30 is 1 per 1,000. Fred 40 is 1 per 10,000. Most sequencing files include per base quality scores and examine them. A smooth Fred profile that rises quickly from the primer region, plateaus through the mid-red, and falls gradually at the end is what clean chemistry produces. A jagged profile, an early drop-off, or a Fred collapse after a homopolymer run each indicate a specific upstream problem.
If you see double peaks at multiple positions throughout the read, two colors overlapping at many sites, that is not heterozygosity. That is a mixed template. Rule it out first. A heterozygous position produces a double peak at one site in an otherwise clean read. A contaminated or pooled template produces double peaks wherever the two sequences diverge. Different biology, same appearance. Check your sample prep before you call a variant.
It is mildly significant that contamination and genomic heterozygosity produce an identical chromatogram signature.
I think mine might be a mixed template. There are double peaks at many positions.
How old is the colony you used for the mini prep?
The plate has been at room temperature since March.
It is June.
I am aware of the month.
Pick a fresh colony from a plate transformed this week. A March plate in June has accumulated satellite colonies and mixed populations. Your mini-prep pooled DNA from multiple bacterial variants. The sequencer is reading all of them simultaneously. That is not a sequencing problem. That is a colony problem.
Bacteria on an agar plate at room temperature continue dividing slowly and accumulate mutations over weeks to months. Plasmid instability, spontaneous deletion events, and satellite colony growth all contribute to population heterogeneity. A mini prep from a plate stored several months at room temperature may contain cells carrying multiple plasmid variants. The sequencing reaction reads all template molecules simultaneously. Every position where variants differ appears as a double peak. The chromatogram is accurate, the sample was not.
Sanger is the confirmatory standard, and it is not a legacy role. After next-generation sequencing identifies candidates, SNPs, indels, small variants, Sanger confirms them one by one. Clinical diagnostics still run Sanger for single-gene panels, BRCA1, BRCA2, CFTR, pharmacogenetic variants. The regulatory pathway is established, and the data is auditable at the base level. In research, every plasmid cloning workflow should end with a Sanger read before the construct goes into cells. Microbial identification by 16S RNA, species bar coding by COI gene. That is Sanger. It is the instrument that checks the other instruments.
The reason Sanger retains its confirmatory role is interpretability. Every base call in a chromatogram is visible, traceable, and attributable to a specific peak with a specific quality score. When a result needs to be explained to a regulatory agency, a clinician, or a committee, the data can be walked through position by position. That level of direct interpretability is not replicated by statistical base calling models derived from NGS data. Sanger's transparency is not a technical constraint, it is a feature.
The global Sanger sequencing services market is running at over 600 million euros annually, not growing at NGS rates but not contracting either. The technique has found its exact ecological niche, confirmatory, single variant, auditable, and it is not being displaced from it.
It is mildly notable that the gold standard genetic reading method works by intentionally stopping DNA synthesis at random positions.
Right, quiet. I need to say something. I have been running Sanger sequencing in my shed for 3 weeks. 72 samples, COI barcoding region, mitochondrial cytochrome, C oxidase subunit 1 from cheese samples purchased at the Saturday market. 6 stalls claim their product comes from specific heritage cattle breeds. I am sequencing the species origin of the milk. 4 of those stalls will be fine. The other 2 are going to have a conversation with me.
Cleanup for batches 4 through 8 is complete. All samples are resuspended and at -20.
Patar has been running precipitation cleanup since last Tuesday. She has processed 48 samples across 2 days.
It is mildly industrious for a consumer protection initiative.
Here is the last thing you need. Your primer placement determines your readable window, and the window is not negotiable. Bases 1 through 50 after the primer are always unreliable. Dead zone, noise, unresolved signal, regardless of template quality or cleanup. Your usable data starts around base 50 and runs to approximately base 700. If the mutation or insert region you're trying to confirm falls within the first 50 bases of the read, you will not be able to call it. Design your primer at least 50 to 100 bases upstream of your target. If you are using a universal primer, M13, T7, SP6, check the primer to insert distance for your specific vector. If the insert starts in the dead zone, order a custom primer positioned further upstream. That is not an extra step. That is primer design. We can read a sequence now, and you know exactly where in that read to trust it and where to throw it out. Next Monday. What happens when one sequence is not enough? Next generation sequencing. Method